So let's move on to some examples. Many people, many scientists in biotechnology and biomedical area wishes to use alginate as beads or capsules. So how to make a bead from alginate? Many of you have probably done this, or at least some of you. We have sodium alginate solution, perhaps about 2%, and we can put cells or particles in this solution and make a drop and drip into calcium chloride solution. So the moment it hits calcium, it will react. And in a few seconds, we will form first a shell, and after some minutes, the calcium will diffuse in, and we get a gel throughout. And then you can pick this out and hold it in your hand. So if you have cells here, you will get a calcium alginate gel bead with entrapped cells. Many types of cell can be entrapped in this way. Bacteria, fungi, human cells, animal cells, plant cells, or something else. So here you see this figure again, how we do it in practice. The blue color is just to illustrate. We added some blue ink. So you can see immediately that in contact with calcium, the bead is formed and the ink will not leak out. We can take this one step further. We can do a coating. There are many reasons for doing this. One reason is that the coating makes the gel stronger. So if we take the bead and let it drip into a polycation solution, and by polycation I mean long chains having positive charges like poly-L-lysine or chitosan. I will talk about chitosan later. These polycations react with the alginate surface. So, and in this way, we get a polycation coating. And interestingly, at the interface between the alginate in blue and the polycation in red, we form something called a polyelectrolyte complex. So negatively charged alginate reacts with positively charged polycation and can form a kind of a film or a membrane. So now this is a much more stable system than we had before. We can go even one step further if we like. We can dissolve the alginate core to make a capsule. And in that case, we put the capsule, we put the bead into a calcium chelator, a compound that binds calcium and picks out the calcium from the bead. And that could be EDTA or EGTA, there are many of those. And quite rapidly, the inside, the blue alginate, dissolves. But the membrane, polyelectrolyte complex membrane, is intact. The reason for doing this is that some cells like it better when it's liquid inside. That's the reason. This depends on the cell type, whether you would do this or not. So the result is we have a capsule with a liquid core and an alginate polycation membrane around it. And this one is stable. I mentioned in the last lecture something I had read in the newspaper, and I'd like to show it to you. This I read on Japan Times Online, 16 October, from Tokyo. This is from a restaurant, not a laboratory. And it says the chef, Jeff Ramsey, doesn't sound Japanese to me, probably some European or American. He demonstrates his molecular cuisine, preparing caviar. And actually, the article described what he had. It said he had syringes like this, I don't know how many, 10 or so, filled with carrot juice, are slowly pressed so they release drops into a clear liquid bath below. We know, of course, what's in that bath, forming bright orange globules. Can you see orange beads? The juice has been blended with a seaweed-derived alginate. The bath below contains nigare. You know that? All Japanese know what that is? I didn't. No? Yeah, that's what it says. It's calcium chloride, a coagulant used for making tofu. This forms a thin membrane around each drop. The result? Instant ikura, salmon caviar, that has never seen a fish. So, probably it's very expensive. But this is today's restaurant, so alginate is on its way into the restaurants. And probably you can make a lot more different dishes in this way. One reason to use alginate is also you can cook it afterwards without melting. So if instead of alginate he had used the collagen or gelatin and afterwards cook it, it will dissolve. But alginate gel will not dissolve when you cook, when heating. And this is just another example of, we think it's alginate. This is from a shop in Kyoto, a supermarket in Kyoto I visited. My colleague said, this is probably alginate beads used for growing plants. How could we tell it was alginate? Well, it was a guess, but alginate gel with calcium has a very typical brittleness. So brittle meaning you squeeze, it breaks in small pieces if you squeeze too much. And if you try to chew, it will also break in small pieces. It will not melt. We didn't do that in the shop, but we looked at it and we took one up and squeezed a little and put it back. So here is another example where we have used an alginate bead to grow plant protoplasts. This is, I think, Arabidopsis. And you see that the cells are dividing and actually grow out of the bead. This was part of a study to study immobilized protoplasts. So you can immobilize almost everything in alginate.